Yeah, thank you very much, Mr. Chairman, ladies and gentlemen. It's a great pleasure for me to speak here at the Exploratory Clinical Development World in London. Understanding the importance of preclinical studies for clinical success. That's the theme of my talk, and in the next 20 minutes or so, I will try to cover some of the aspects which you can see here. I will briefly touch on the loss of R&D productivity. I will show you some examples from our own pipeline where compounds failed in research and development. I will try to discuss some of the lessons learned, and I will try to present what we believe are opportunities for better investigations and opportunities for cross-functional cooperations. If we look at the development of the global R&D expenditures, the development times and the output of new molecular entities over the last couple of years, and I'm very sure that you all know these figures, it is very obvious that the development costs are still rising and at an alarming rate. In fact, they have doubled over the last decade. The duration of development is increased, and that there is, or there appears at least, to be a decreasing success rate for new drug candidates. With other words, and this is something which we certainly had to learn, industry is spending more and getting less from current drug development efforts, and this development is called or perceived as the loss of productivity. If we now have a closer look at the probability of technical success in the different phases of the development process, it is clear that the likelihood of technical success of an individual project moving into development is pretty low. In fact, at the start of the preclinical development, each drug candidate has a statistical probability of technical success of only 5%. If we translate this figure into number of compounds, it is very clear and obvious that we need 20 drug candidates at the beginning of preclinical development in order to have the chance, the statistical chance, to bring one drug to the market. It is also obvious that most of the candidates are failing in the early phases. Of the 20 candidates which are entering preclinical development, 13 are lost during the preclinical phase and during the initial clinical phase. But importantly, of the 7 candidates which are entering larger and certainly very expensive clinical trials, only one drug will finally pass all hurdles and reach the market. And these late failures are consuming a lot of resources and costing a lot of money. This situation poses a very significant challenge, in particular to the early development process. As a consequence, pharmaceutical companies are re-evaluating their strategies to redesign R&D to make more effective and informed go-no decisions as early as possible. And part of this strategy is to eliminate those drug candidates which will not make it through development early on, and by doing this, we have to identify the candidates with the best chances of technical success to be tested in our very limited clinical resources, or with other words, we have in these early phases to manage the failures. If we have a closer look at the reasons for failure in drug development, you can see that a comparably high number of terminations is still due to non-clinical findings, pharmacokinetics, and especially non-clinical toxicity findings. And in fact, up to a third of our development candidates are abandoned because they are causing toxicities in laboratory animals. Going into more detail, the kinetic reasons for failures in drug development include a short half-life, low bioavailability, risk for interactions, or the detection and identification of reactive metabolites. The toxicological aspects include a narrow therapeutic window in relation to the intended indication or in connection with the potential for drug interactions. Critical findings such as genotoxicity, liver toxicity, cardiotoxicity, And what I feel is probably the most important aspect, our inability to clinically monitor certain toxicities. This clearly underlines the necessity for predictive tools which allow an early assessment of the potential liabilities of a new drug candidate. In the following two charts, I will briefly show the screening cascades which we have in place in order to characterize the PK and toxicological properties of new drug candidates. It may not be necessary to go into much detail because I believe that they are more or less comparable in each of pharmaceutical companies. Here with the DMPK screening cascade, we are starting usually with some in vitro investigations confirming the in vitro results under in vivo conditions, and finally making our PK/PD modeling predictions about the dose to be used in the clinical trials, and also about the possible safety margin. In toxicology, we have a very stringent screening battery, which usually starts with an in silico prediction, which means that we are using computer programs to investigate questions like, are there structural alerts, or can potential toxicities be predicted based on the mode of the action of the new compound. During the first safety assessment, we are trying to figure out liabilities which can be very easily tested. This is, for example, a genotoxic liability, which can be easily detected in an Ames or in a cytogenetic in vitro screen. We have to exclude or investigate the liability of a new drug candidate for cardiotoxicity, and this is usually being done in a Herc assay. And in this phase, we are usually doing a first limited in vivo study, which is normally a limited rat screen. And then we are moving into the exploratory toxicology for the definite candidate, and this exploratory toxicology program usually includes repeat dose toxicity studies in 2 animal species, safety, pharmacology. And with this, we are coming then to the decision whether or not to move a compound into development. And once we are in development, we have to establish the safety of the drug candidate before giving it for the first time to humans. This is in this phase prior to first human exposure. And later, we have to support the full clinical development. In the next couple of minutes, I will present some examples of toxicological reasons for failures of development candidates. And I will discuss when these findings leading to the termination of the project were detected during development. It is clear that this is our own history from the company where I am working. Each company may have its own history, but I want to use these examples to derive some general principles from them. And it is certainly also not necessary that I go into the details of each of these cases. The structure of the table is always the same. Here on the left side, you see the 5 candidates, which are named candidate 1 to 5. And at the top, you can see the toxicological programs which have been performed, the 2-week studies, which usually is representing the exploratory phase, then the 4-week studies, which are representing the GLP or IND-enabling program to support the first human dose, and then the studies 13-week, 6 months, and so on, which are supporting the full clinical program. The dotted red line marks the transition from the preclinical to the clinical phase. So let's have first a look at the findings. Candidate 1 was terminated after a severe vasculitis was observed in the coronary arteries of dogs. Candidate 2 was terminated after we got a very strong evidence of liver toxicity in dogs. And in addition, in rats, we got signs of a hyperplasia in the urinary bladder. The candidate 3 was terminated after we found necrotic lesions in the pancreas of dogs. Candidate 4 was terminated after tubular degenerations were observed in the kidneys, both in rats and in dogs. And the candidate 5 was terminated after mammary tumors were found in rats. And as you can see from this table of the 5 candidates which I'm presenting, 4 were terminated due to findings in the 4-week tox studies and before humans were exposed to the drug candidates. One candidate number 5, was terminated after it had entered clinical development. So looking at this chart again, one may come to the conclusion that at least 4 projects were terminated early on and before larger and resource-consuming programs were initiated. The question is, however, whether there was a chance to detect these findings even earlier, and this is shown here in this slide. You can see that for 3 candidates, and this is candidate number 1, 3, and 5. There were no signals in the preceding toxicological studies, and this explains here the color green, so no signal. Whereas for candidate 2 and 4, a slight signal indicated by the color yellow was already observed in the 2-week toxicity studies. There is reason to believe that it might be possible to detect such findings even earlier. We saw it in the 2-week toxicity studies, but I think there is good reason to believe that we can even detect them earlier. And this can be achieved if we are combining our traditional toxicological studies, and I will come to this in a minute, with more novel technologies, and thereby might be able to further improve the strength and the power of our screening cascade. And this could occur here at these 2 levels. In fact, if we would only rely on the established parameters which we have used over many, many years, clinical observations, clinical chemistry, hematology, and histopathology, an earlier signal detection may not be possible. That's very obvious. As an example, for many years, only serum creatinine and blood urea nitrogen were used, for example, to measure kidney damage. And these 2 biomarkers are not very accurate. They are not very sensitive, and they are indicating a possible kidney problem far too late.